Cell cycle. Let us we will discuss about the cell cycle. Introduction. Cell cycle, the order of sequence of event that occurs in the cell in the preparation for the cell division. The cell cycle is 4 stages process in which the cell is increasing in the size. Gap phase or G1 phase copies its DNA. Synthesis or S phase prepared to divide. Z2 stage or gap 2 and divided into mitosis or M stage. Z1, S and Z2 make intercones for the span between the cell division. Here we will see that mitosis. Mitosis is the process where a single cell is divided into two daughter identical cells. And meiosis is the process where the single cell is divided into twice to produce a 4 cells. Here we will see that cell cycle. Cell cycle is divided into interphase and mitosis or M phase. And interphase is divided into Z0, Z1, S and Z2. And mitosis is divided into prophase, metaphase, anaphase and telophase. The cell cycle is divided into two phases, interphase and mitosis. Here we will see the cell cycle. What happened the cell cycle and how it is divided into how many types. Let us we will see. Here we will see that the cell growth. The cell growth is occurring. Cell growth occurs in the organelle cells and double. Z1 we will see the checkpoint and cell produced the proteins and enzymes. Growth phase and length period is 5 hours and cellular division is occurs. And next we will see the Z0 phase, cell generating or resting period. And next we will see that the S phase. S phase is nothing but a synthesis phase. Here we will see the DNA synthesis and replication is occurs. Period of time is 7 hours and post synthesis phase. And it is about 2n condition to 4n condition and growth occurs in the Z0. And here pre-mitotic phase and cell growth and cell replication is occurs and the length of the period is 3 hours. And here we will see that the M phase. M phase in that the mitosis and cytokinesis. Let us see. In mitosis we will see that prophase, metaphase, anaphase and telophase. After last cytokinesis is occurs and it is produced the two daughter cells. And Z0 phase. The cell cycle matching is dismethylated and cyclins and cyclin depending kinase as disappears. Cell then remains in the Z0 phase. Other type of the cells such as epithelial cells continue to divide throughout an organism's life and rarely enter to Z0. S phase. Phosphorylating or activating protein enzymes. Z1. The cell cycle grows in the cell synthesis mRNA and protein that are required for the DNA synthesis. Once the required the proteins and growth are completed, the cell enters into the next phase of the cell cycle, Z2 phase. The cell has grown, DNA has been replicated and now the cell is almost ready to divide it. This is the last stage is all about the preparing the cell for mitosis. Cytokinesis is the physical process of cell division which is divided to cytoplasm and parental cell into two daughter cells. Karyokinesis is the division of nucleus that occurs in the 4 stages. They are prophase, metaphase, anaphase, telophase. During prophase, chromatin condenses to form chromosome. Interphase, prophase, metaphase and anaphase, telophase and cytokinesis. In interphase, chromosomes are arranged like a thread-like manner and they are so clumsy. In that prophase, they are divided and moves towards. In metaphase, they are packed with each other and divided into two identical chromosomes, and that anaphase they are moved to give some force to divide it, and that telophase is occurs. The two daughter cells, some notches are occurs in the cell, and that cytokinesis is occurs. So the cells are divided, and that see here what happened. Pre-synthetic phase, S phase, DNA synthesis phase. Z2 phase is a pre-mitotic phase. In interphase Z1 as Z2, Z1, Z2 get to growth and ready to synthesis the S phase. In mitosis is divided into 4 phases. Already we will discuss the prophase, metaphase, anaphase, telophase. So prophase is the first stage to mitosis process that separate the duplicated genetic material carry in the nucleus of parent cell into two identical daughter cells. During prophase, the complex of the DNA proteins contained in the nucleus is known as a chromatin condense. Metaphase is the stage of mitosis in the eukaryotic cell in which the chromosomes are at their second most condensed and coiled stage. Anaphase is the stage of mitosis after the process of metaphase, when replicated chromosomes are split and newly coupled chromosomes are moved to opposite poles of cell. The next, telophase is the final phase of mitosis. The process separate the duplicated genetic material and carry in the nucleus of parent cell into two daughter. Telophase begins to once the replicated paired chromosomes have been separated and pulled into opposite sides or poles of the cell. Interphase. During this phase, the cell grows, accumulating the nutrient needed for the mitosis, duplicating its DNA. Mitosis, during which the cell is split into two distinct cells. Checkpoint. DNA damage checkpoint. This DNA damage both before the cell enters into the S phase as well as after S phase. Damage to DNA before the cell enters into the S phase, inhibiting the action of the CDK2, thus stopping the propagation of the cell cycle until the damage can be repaired. If the damage so severe that it cannot be repaired, then cell death by apoptosis. Cell cycle checkpoint. These are cell cycle control mechanisms in eukaryotic cells. These checkpoints verify where the process at each phase of the cell cycle have been accurately completed before progressing. See here there is three main checkpoints: Z1, Z2 and metaphase checkpoint. Here we will see that Z1 and S, M2, M checkpoint, Z2 checkpoint. S is the synthesis phase. We will see that Z1 checkpoint, the restriction point of mainly controlled by the action of the CKI P16 CDK inhibitor P16. The CDK not binded to the cyclin DI active cyclins D. CDK complex phosphorylative reticuloblastoma protein in the nucleus. Point. Maturation promoting factor. M-phase promoting factor is a protein composed of cyclin B and CDKI. This protein promotes the Z2 phase. Entrance of M-phase. Trigger the formation of mitotic. Promote the chromosome condensation. Causes the nuclear enveloping blockage. And metaphase checkpoint. This occurs at the metaphase, anaphase, promoting complex regulate checkpoint. This checkpoint whether all the chromosomes are properly attached to spindle or not. Next, cell cycle is regulated by the cyclins, cyclin depending kinase, that is called a CDKS, and cyclin depending kinase inhibitor, that is CDKIS. Cyclins. Cyclins are family of protein which is regulated the cell cycle. There are several types of the cyclins that are active in the different parts of cell cycles and causes the phosphorylation of CDK. Cyclins are mainly types cyclin A, cyclin B, cyclin C and cyclin D. Cyclins derive the events the cell cycle by prenating with a family of enzymes called cyclin depending kinase. Alone CDK is inactive, but binding to the cyclins activate it makes the functional enzymes and allowing it finally modified to target the protein during estrase. Cyclin A2 is mostly located in the nucleus and it regulated the inhibition and propagation of DNA synthesis. Play an important role to control all the cell division. Cyclins that are provided to domain essential for the enzymatic activity through phosphorylation CDKS signals the cell that it is ready to pass into the next stage, the cell cycle. Cyclins depending protein kinase or depending on the cyclin binding to CDKS activating the CDKS to phosphorylating the other molecule. They are present in the eukaryotic animals containing at least CDK4 of which CDK1, 2, 3, 4 are directly involved in the cell regulating in mammals cells. CDK1 with a partner cyclins A2 and B1. Another one CDK7 is indirectly involved in the CDK activating kinase. Cyclin binding. The activity site or ATP binding site or all kinase is cleft between the small amino terminal lobe and large carboxy terminal lobe. The structure of human CDK2 relieved that CDK have modified the ATP binding site that can be regulated by cyclin binding. Is that way to cyclic process to divided the one cell into another cell and it is the process to cell division and that is called as a cell cycle. Cyclin binding. The activity site or ATP binding site or all kinase is cleft between the small amino terminal lobe and large carboxy terminal lobe. The structure of human CDK2 relieved that CDK have modified the ATP binding site that can be regulated by cyclin binding. Is that way to cyclic process to divided the one cell into another cell and it is the process to cell division and that is called as a cell cycle. That cell cycle mitosis, and in that mitosis what we will see here, the cell is resting period, it is called as Z0 and that resting period is duplicating generation is starting, in that replication of DNA is occurs and next S it is the synthesis phase. In that synthesis phase the 2n condition or 4n condition post synthesis is occurs. In that S is further it divided in, it is converted into Z0 pre-mitotic phase and that cell preparing to check the what the cell is going to generate what the cells. In that the Z2 is divided nucleus in the form the mitosis. In mitosis we will see the prophase, metaphase, anaphase and telophase. In that cytokinesis is occurs. In that two daughter cells are formed. Let us we will see. Z1 is containing the 5 hours time and it is the length of the period. Next, S is 7 hours and Z2 is 3 hours and mitosis is 1 hours. And that tightly chromosomes are arranged in the prophase, and that metaphase is two opposite poles linearly mitotic spindle they are divided. In that anaphase, breakdown is occurs and moves to positive opposite poles. And next, telophase they are getting to divide the cells, and that cytokinesis is occurs. In that cytokinesis, there are two daughter cells are formed.